school's out, blame corona, but I still got some science I could show ya. No class, no school, that's cool, throw the class on the YouTube. Even if they shut the school down, I'll get that knowledge anyway somehow. Real scholars won't stop for corona. Tune in for that science takeover.
Happy Monday, everybody. Welcome to Mrs. Finney's science class. Shout out to our HB Warriors, to all of our subscribers, and to all of our visitors. Today, we are tackling a new lesson, and our lesson is lesson 8, proteins, genes, and chromosomes. Actually tackling a new section, section 2, structure of proteins and resulting traits. Learning objective today. I can explain what happens if the shape of a protein changes. Does it matter? Will anything change? Can it still do its job? We'll see today. All right, so I want you to take a look here at investigation 1 within that particular section. It's titled Modeling the Gene to Protein to Trait Relationship. There's an image in here. It's a bit odd, but check it out. Don't be alarmed. So here we have an image of glow-in-the-dark pigs. Yes, glow-in-the-dark pigs. What single topic in biology has led to technology that makes glow-in-the-dark pigs? The ability to know your ancestral history and the production of real-life clone sheep. All right, that topic is the study of genetics. And today, we're going to take a look at a video that gives us a little more insight as to how in the world can we have glow-in-the-dark pigs. Really? Glow-in-the-dark pigs? Yes, it happened. Take a look here at this video.
The world's first glow-in-the-dark pig after injecting them with jellyfish DNA. I'm Michael McCrudden talking to you about glow-in-the-dark piglets here for you on I.O.
Hey Mike, you can't wear that shirt.
This is a Christmas present and I promised I would wear it.
It messes up the green screen.
When I opened it up this Christmas, I said I will wear this on I.O. I love it. And I'm doing a story about glow-in-the-dark pigs, so it's kind of fitting. Back to the story. We're talking about these Chinese scientists who messed around with some pigs, and now when they turn off the lights, these piglets, they emit a green fluorescent light. Now there were 10 pigs who were injected with a jellyfish DNA, and all 10 of them are happy, healthy, and scientists, they believe they will go on to live a normal lifespan. That means they have a few years ahead of them, and it's unlikely they'll end up as a sausage McMuffin at McDonald's, unless McDonald's starts tinkering with glow-in-the-dark food. McDonald's food probably already glows in the dark. Now if you feel bad for the pigs, they are not the only animals getting injected with jellyfish DNA. In Hawaii and in Turkey, they also did it to rabbits, they've done it to mice, they even did it to kittens, but the kittens, instead of glowing green, they glue red. You might be wondering, what's this all about? Well, the green tint, it allows the scientists to know that their injections have actually bonded with the natural makeup of the animal. So they can go and throw in stuff into the embryos, and it is going to multiply and grow and become part of the animal. And from there, the ultimate goal is to start introducing beneficial genes into larger animals, and they can start growing natural, healthier embryos, which will save a lot of money on creating medicine when they can just grow healthier, happier creatures.
How shocking is that? Glow-in-the-dark pigs. How do you feel about it? Please drop your comment in the section below. All right? Please let me know. What do you think about glow-in-the-dark pigs? Interesting discovery. All right. So here in this investigation, it says studying genetics gives us the ability to know how traits like glowing in the dark form, how they have changed over time, and how we can change them now to help people. All right. Click on the buttons to learn how each story is related to DNA. We talked about DNA, right? Take a look here. Do you know your ancestry? People have been moving around the globe for thousands of years. We can learn a lot about who our ancestors are by analyzing our DNA. Several companies offer DNA analysis to let you know what part of the world your ancestors come from. All right, so that is one key piece about DNA. We just looked at a video that talks about this. These pigs are glowing because a gene from a glowing jellyfish was added to the DNA of a pig while it was an embryo. As the embryo grew, it made lots of new cells that had the jellyfish gene inside of them. With this gene, the pig cells make a protein that makes the pig glow. And then last but not least, Dolly the sheep looks like a normal sheep, but she did not have a normal beginning. Scientists fused the nucleus of a sheep's udder cell with a sheep egg cell whose DNA had been removed. They then implanted the egg cell with its new nucleus into a surrogate mother who gave birth to Dolly. All right, so these are just 3 different pieces of genetics that I wanted to show you guys related to DNA. All right, we'll take a look here before we get into our next piece. So DNA, genes, and chromosomes are probably words you have heard before this lesson. We've definitely talked about them. All right, and what have you already learned about DNA and where it is located? Drop that in the comment section. What have you heard about genes? You can answer that as well. What do you know about chromosomes? Okay. What kinds of professional work depend on understanding genetics? Brainstorm ideas in your notebook and then discuss. All right, so this is just an illustration of chromosome, and then the DNA wraps around it, and then we have genes on the DNA. All right, I want to take you to our piece in section 2 that gives a little more information about the structure of proteins and resulting traits. If you cut off all of the prongs on a fork, just imagine that. Don't go in your kitchen and do that. So that it looked more like a spoon, would it still function as a fork? No. The structure of an object, especially a tool, is very important for its function. So what happens if the shape of a protein changes? Do you still think the function will be the same? Let's take a look into a little bit more information here. The shape of each kind of protein is unique and helps determine its function. One small change to the shape might cause the protein to fall apart or not be able to do its job in the cell anymore. For example, there's a protein called hemoglobin that is found in red blood cells. Hemoglobin is an important protein because it carries oxygen. It's a protein that carries oxygen. What is it called? Hemoglobin. One more time. Hemoglobin. All right. It carries what? Oxygen. All right. Learn something new every day. Hemoglobin is an important protein because it carries oxygen. If the shape of the hemoglobin protein changes in a certain way, the shape of the red blood cells can also change. Instead of a blood cell being round or disc shaped like this one, it becomes bent and crescent shaped like this one. These mishapen cells are called sickle cells. This one right here. Carrying oxygen through the body is an important trait for survival. We talked about that. And sickle cells are much less efficient at carrying oxygen in the blood. In addition, they can get stuck in narrow blood vessels because of their shape. Now let's take a look at 2 videos to give you a little more information about how this happens.
Our red blood cells make up 40% of our blood's volume. They're made continuously in the bone marrow, looking like flattened donuts without the holes. Red blood cells contain hemoglobin, a bright red protein which gives blood its color. When we breathe, as our blood passes through our lungs, oxygen molecules attach to the hemoglobin. Then, the red blood cells and hemoglobin transport the oxygen throughout the body and release it to all our cells and tissues. When the hemoglobin molecules are emptied, they attract carbon dioxide and other waste gases, which they take back to the lungs to be exhaled. Anemia occurs when there are too few red blood cells or not enough hemoglobin. Exhaustion is the common sign of anemia.
All right, so now we know the role of red blood cells in sickle cell anemia, right? Let's look at another video that gives us some information about the history of sickle cell.
Imagine for a minute that your blood cells are shaped like sickles, sort of like that thing that the image of death carries. Ouch, right? But what exactly causes our friendly little red blood cells to turn against us? Sickle cell anemia is a blood disorder where instead of supple, soft, frisbee red blood cells that live for 120 days, your body forms stiff, pointy, sickle-shaped cells that only live for 10 to 20 days. RBCs need to be soft and pliable to squeeze through your blood vessels safely and efficiently. So when they're pointy, they can get stuck in there. When this happens in the chest, abdomen, or joints, it can cause pain. If they get stuck in the hands or feet, you can get swelling. And of course, if they get stuck in vital organs, they can cause infections, or if they get caught in the eye, vision problems. And if that wasn't bad enough, the brittle cells can break apart, delaying oxygen delivery and causing fatigue. This all sounds terrible. It was first described in 1910 in a dental student named Walter Clement Noel, who went to a doctor in Chicago complaining of pain. But this student wasn't from Chicago, he was from Grenada. Sickle cell anemia predominantly affects approximately 1 in 400 African American births. It was a disease known in Africa for about 5,000 years, but it had never been described in Western medicine before about 100 years ago. The doctor in Chicago, whose name was James B. Herrick, was the first to describe it in a paper, and over the next century, many doctors have attempted to uncover just how this debilitating disorder works and why it mainly affects those of African descent. In the 1930s, a DC-based doctor named William Cardozo was one of the first black doctors to conduct research into this disease, and he did so with a grant from Alpha Phi Alpha, the first black fraternal organization. Cardozo felt that research into sickle cell anemia had, quote, reached an impasse, and that no one had yet found the cause of this horrible disease. If someone could provide a new stimulus to get the research going, he wrote, maybe we could discover more. His research, published in 1937, helped medicine realize that sickle cell anemia was inherited. Thanks to his research, we now know that sickle cell anemia came from us. And he helped explain why it mainly affected people of African descent, which is huge. While he was working on that, other doctors started to realize that this disease was based on oxygen content in the blood. In fact, Quebecois researchers in 1930 Montreal took a 7-year-old black girl and restricted blood flow to her finger with a rubber band. This probably wouldn't pass an ethics board today. In doing so, they found, though, that sickle cells formed in the low oxygen environment of her fingertip. They learned that with a bit of hypoxia, or lack of oxygen, the normal cells could collapse into all manner of weird and pointy shapes, nearly instantly returning to normal when exposed to air. Their methods were questionable, to say the least, but the results did shape future research. In the mid-20th century, doctors discovered that a single amino acid was responsible for the hemoglobin on the red blood cells that caused the sickling. But now that we understand the disorder enough to try and solve it, unfortunately, we're at another moment when we may need more new stimulus, like Cardozo said. According to a clinical investigation from 2007, there are many possible treatments for this disease, including a bone marrow transplant, but there is still no cure. One option is medicine that helps fetuses develop more hemoglobin, but it can make the disease worse. Maybe in the future we could get genetic manipulation of the systems that help our bodies create blood using genome engineering or gene therapy. But for now, sickle cell anemia is just out there, a life-threatening thorn in many people's sides. Cardozo was one of many doctors of many races in the hunt for causes of sickle cell anemia. He's notable because in the 1930s, doctors that looked like him were often not welcome in the halls of medicine. That he was able to make a discovery for a disorder that affected so many people that also looked like him is a pretty big deal. So we're happy to recognize the part he played in this story for Black History Month. For more on red blood, watch Jules' video about what bone marrow actually does here. Hint, it is very important. And please subscribe for more Seeker. When I think of red blood cells, I think of those soft little frisbees we used to throw around as kids. I used to hit my brothers with them. Maybe that's why I think of RBCs as just so darn friendly. Thanks for watching.
All right, so that was a lot of information to give you an understanding of sickle cell anemia, how that happens, and it's due to sickle cells. Let's have a flashback of some information that we reviewed before watching those videos, okay? And so there's a protein called hemoglobin that is found in red blood cells. Hemoglobin is an important protein because it carries oxygen, okay? If the shape of the hemoglobin protein changes in a certain way, the shape of the red blood cells can also change. So that is the case in sickle cell disease, all right? So let's take a look down here a little further. It says, in the way that a fork with no prongs might still work as a spoon, the change in a protein structure could also result in a slightly different function for the protein. For example, there is a protein that helps make up part of the taste buds on the tongue. Okay? This protein helps a person taste different substances. I'm so glad for that because I love to taste different foods, don't you? What do you really like to eat? It's like something you had at any point that you think is delicious. Please drop that in the comments section. Shout out to our taste buds, right? And shout out to that protein that helps us make up part of taste buds on the tongue. This protein helps a person taste different substances. Differences in the shape of that protein allows some people to be more sensitive to the taste of bitter foods or drinks. You might avoid bitter foods if you have this version of the bitter taste protein. Go figure, right? So let's take a look here at our check for understanding section. Drag the terms to their appropriate locations. Word bank. The same, unique, a different, or similar. Which one should we place? And the box is right here. Okay, let's figure this out. The shape of every protein is blank and helps determine its function. Which word do you think we should pull? Unique? Let's see. When a protein structure changes, it will likely perform blank function. What do you think? A different? Let's go for it. Let's go. Drag and drop. Nice job. If you had this right, give yourself a round of applause. If you didn't, give yourself a round of applause just for trying. Shout out to you. All right, so it says the shape of every protein is unique and helps determine its function. When a protein structure changes, it will likely perform a different function. All right. We have 2 questions here I want you to think about before I show you the response. Why does the shape of a protein matter? In your own words. Yeah, I'm waiting for you. Yes, I am. All right, so right here it says the shape of a protein determines its function. All right, I think we have that down pack for today. Down pack, yes, down pack. Yes. Okay, next one. What protein changes shape in sickle cell anemia? What are 2 effects of this change? What's your answer? Yeah, go ahead. Okay. Hemoglobin changes shape. This changes the shape of blood cells and makes them less efficient at carrying oxygen. It also makes them get caught in narrow blood vessels. All right? So the protein that changes shape is hemoglobin. All right? Nice work. Make sure you jot these answers down in your TCI program. Again, we're on lesson 8, section 2. Go inside of your notebook and make sure you have these 2 responses in there for today. All right? And you can put those in your own words if you like. All right, so let's go back here. Make sure we have our essential question answer. Learn objective. I can explain what happens if the shape of a protein changes. Right? Right? Drop that in the comment section. Okay? Your answer to this essential question. What happens if the shape of a protein changes? What would you say? Leave that in the comments. All right? So the answer, the shape of each kind of protein is unique and helps determine its function. One small change to the shape might cause the protein to fall apart or not be able to do its job in the cell anymore. So in your own words, please express your answer to this essential question in the comment section. All right, and we're going to close out for today. Close out for today. Close out, close out, close out, close out, close out, close out. I used to be a cheerleader and a cheerleading coach, so it comes out of me every now and then. Woo, woo. All right, here we go. Let's close out with our outro. It's been fun. It's been real. We break it down and pick up science skills. Give yourself 2 claps just for tuning in. Come back, meet tomorrow. We can learn again. Oh, it's been fun. It's been real. We break it down and pick up science skills. Give yourself 2 claps just for tuning in. Come back, meet tomorrow. We can learn again. I will see you all tomorrow. Have an awesome rest of your day. Peace.